This is the lab video for Experiment 9, the reaction of iodoethane with sodium saccharin. In preparation for this chemical reaction, I've set up some glassware in the hood. I have a hot plate stirrer. On that hot plate stirrer, I have a thermocouple plugged into it that gives feedback into the hot plate to control the temperature. I've set the hot plate to 80 degrees and it's holding it somewhere between 79 and 82 degrees, which is close. I have this hot plate set to stir at 300 RPM, and I'm going to use the timer on it when we're ready to go. On top of that hot plate, I set up a water bath. So it's been heating now for a while at 80 degrees C. Sitting inside that water bath is a 125 Erlenmeyer flask with a stirring bar in there. It's a nice, pretty red one. We're now going to start adding our chemical reagents. The first thing I'm going to do is go out and measure out approximately 10 millimoles of sodium saccharin. Come back, add that to the flask, and then we'll add the rest of our reagents. Here is the container of sodium saccharin. I've taken it over to the balance and I've weighed out approximately 10 millimoles. And here it is in this weighing paper. I'm going to add it now to my Erlenmeyer flask. To that, I'm going to add some dimethylformamide as our reaction solvent. Here is my dimethylformamide from the chemical stockroom. I've measured out approximately 5 milliliters of that dimethylformamide. I'm going to add that now to my Erlenmeyer flask. And we're going to let that stir until everything dissolves. I'm going to use my glass rod a little bit to help mix it all up. To my Erlenmeyer now, I'm going to add 10 millimoles of iodoethane. It is a liquid, so I'm going to use a pipette to deliver that. I've placed several milliliters of iodoethane into this very small beaker. I'm now going to use a graduated pipette and a bulb to actually deliver 0.8 milliliters of solution into my reaction vessel. And deliver it on in there. Now we're going to continue that. I'm going to set the timer back up to 10, and we're going to allow that to mix for 10 minutes and heat for 10 minutes. Occasionally, I'm going to come back in and just mix things up a little bit. While the reaction is undergoing, I'm going to cover it with some parafilm so nothing falls into it, and we'll continue to let it stir. I'm currently at 7 minutes and 50 seconds and counting down. I'm still around 80 to 82 degrees. We'll continue to wait. It's been just about 5 minutes. We have about 2.5 minutes left. We're down to the final 10 seconds now of our 10-minute reaction time. I'm now going to remove a reaction vessel from the hot water bath and set it on the counter here to cool to room temperature. I'm going to slide the hot plate out of the way, and we'll wait about another 10 or 15 minutes for that to cool to room temperature. While we're waiting, I'm actually going to remove my stir bar from the reaction vessel using a magnet. So I'll pull it out and grab it with this little forceps. Put my parafilm back on the top. It has been about 10 minutes and it's still a little warm to the touch. So I'm going to let it stay there a little bit longer before I go on to the next step. It has been about 20 minutes and it feels like it's at room temperature. The next step in this reaction is to add 40 milliliters of DI water, which I've measured out in my graduated cylinder. I'm going to pour that in. You can see a white precipitate formed. I'm going to transition that over to a ice bath now. And once I feel that the solid has all precipitated out, I will go over to the vacuum filtration system and filter out the solid. So we'll let that stand for another 10 to 15 minutes. It's been about 15 minutes now, and if I look at the reaction mixture, I'm going to try to bring it closer here. You can see there's sort of a white yellow solid suspended throughout the solution and a bunch at the bottom. I'm now ready to take that over to my vacuum filtration system and filter out my solid. Here are my lab notebook pages for this first part of the experiment. So I've brought my reaction mixture over to the vacuum filtration apparatus. I have chilled some water in an ice bath here. I'm now going to take a piece of filter paper, place it into my Buchner funnel, turn on the vacuum, and I'll take some of that chilled water just to seal the vacuum paper to the bottom. Okay. And then I'll take my reaction vessel and pour the contents down into the Buchner funnel. I'm also going to rinse out my Erlenmeyer flask with some ice cold water. And we'll do that a couple times until we think our reaction vessel looks clean. And we'll do it a third time here because I can still see product down in that Erlenmeyer flask. Okay, let me set that aside. We're going to let that vacuum sit there and pull on that product. In the meantime, I'm going to go over and pre-weigh a watch glass so that we can actually transfer our product over to a watch glass and then go dry it in a drying oven. This is the mass of a clean watch glass. I brought my watch glass back that I've weighed. I'm now going to take and turn off my vacuum, remove my filter funnel, and scrape the contents onto my pre-weighed watch glass. And then I'll take my filter paper and scrape anything that might still be on it. And there we are. I'm now going to take that back over to the balance, weigh it, bring it over to the oven, let it bake for 5-minute intervals, and then weigh it in between each until I reach a constant mass. This is the mass of the watch glass and our product before going into the oven. Our picture of our product in the oven. After baking for 5 minutes at 90 degrees centigrade. After 10 minutes in the drying oven. After 15 minutes. And after 20 minutes, and the mass is staying fairly constant. Well, I'm back from the drying oven. Now I'm going to prepare a sample for doing some nuclear magnetic resonance. Here is our product. I have it covered with a watch glass also. I have a small vial, some deuterochloroform, a clean pasture pipette, and an NMR tube. I'm going to take approximately 1.5 to 2 milliliters of my deuterochloroform, put into my small vial, and then I will weigh out approximately 5 to 10 milligrams of my product to dissolve into that deuterochloroform, and then put some of that, approximately about 3/4 of a milliliter, into my NMR tube. Let's first go weigh out 5 milligrams. I've placed 6.9 milligrams of our product into the small vial. Okay, I'm back. I've weighed out approximately 5 to 10 milligrams. I'm going to remove the rest of my product. I'm now going to take a clean pasture pipette and dispense in approximately 2 milliliters of my deuterochloroform. There's approximately 1, and there's approximately 2 milliliters. Cover that back up. Take my sample, make sure it completely dissolves. And now I'll take some of our dissolved product and I will put it into the NMR tube using a second pasture pipette. And that's all we need. We're now going to take that over to the NMR spectrometer and we'll run a spectrum of our sample. Here we are in the NMR lab. We're going to submit our sample into the queue, and we'll come back later today and look at the results on the computer. Our NMR sample has finished being run. I've now gone over and I've opened up some processing software so we can look at the spectrum. I'm just going to go up here, and there it is. I'm going to open it up. I'm going to expand it to full scale. I'm also going to expand the scale a little more between 8 ppm and 0 ppm so we can see it a little bit better. If I look over here on the left-hand side, that is an aromatic region. This peak down here is due to our solvent. I can see a quartet, a triplet, and a triplet. We're now going to do a peak select to pick out the ppm shifts for all these peaks. However, I first need to set a baseline so that I don't pick up all these little teeny peaks. So I'm going to set the baseline here. I'm going to grab it and I'm going to move it just above this peak right here so that we can filter those out. And then I'm going to have the software pick all the peaks automatically. Notice it picked out 4 here for this quartet, 4 for this quartet, 3 for this triplet, 3 for this triplet, and a whole bunch load for this aromatic region. Now we're going to go in and we're going to integrate the area underneath these specific regions. So I'm going to come up here. I'm going to select the expansion tool because I need to look at these areas a little bit closer. So let's first look at the aromatic region. I'm now going to select the integration tool and I'm going to integrate from one side to the other. Notice it integrated to 19.04, but we know something special about these molecules is that this area will integrate out to 4, so let's force it to 4 so that this area is 4 and all the other areas that we integrate will be relative to that area of 4. Let me go back and expand the scale back up again. Let's go back in and let's expand the area for the triplets. I'm going to integrate underneath those areas. Go up here, pick up the integration tool.
integrate this area, and I'll integrate this area. Okay. Let me zoom out again. I'm now going to zoom back in to this area. I'm going to pick up my integration tool. I will first integrate this triplet, and I'm going to pick an area somewhere between them. Notice there's a little bit of overlap. In this case, I'm going to select the area and come from the left-hand side to collect that area. And there they are, integrated. So let me zoom out again. I'm now going to go up here to Options, and I'm going to select the layout. I'm going to select 2 columns. I'm going to come up to this tool here, which allows me to highlight certain areas. I'm going to create a new version of just that area. I'm going to create a version of just this area. And finally, I'm going to create a box with this area in it here. I will now include it as a separate PDF file for you to analyze. This is the last page of our lab notebook for this experiment.